Today we're going to discuss how different things can affect the rate of an enzyme-controlled reaction. There are three main factors that are going to affect reaction rate: temperature, pH, and concentration. And remember, enzymes, their primary function is really to speed up a chemical reaction. So we're going to go through each of the three different factors individually and see how each of them will affect the rate of a reaction. The first factor that we're going to discuss is temperature. We're going to look how the temperature affects a chemical reaction using an enzyme. There are a couple of things that you need to remember back to your physical science days. As the temperature decreases, molecules move slower. As temperature increases, molecules move faster. And if you remember, reactions occur due to collisions between molecules. So if the molecules are moving faster, they have a better chance at colliding with each other. If they're moving slower, they have less of a chance of colliding with each other, thus having fewer reactions. So if we look at temperature, the general rules for temperature is that as temperature decreases, the rate of reaction also decreases. This makes sense because we know that as temperature decreases, our molecules are moving slower. They're having fewer collisions, which would logically slow down our reactions with an enzyme. The temperature increasing causes a rate of the reaction to increase as well. There's a trick though. It will also decrease after a period of time. We're going to see why in a second. The initial increase, though, does make sense. As temperature increases, the molecules are going to move faster and collide with each other more frequently, causing the rate of the reaction to go up. Let's explain this next part, though. If we look at our generic graph for how temperature affects reaction rate, the reaction rate increases on the y-axis and the temperature increases on the x. You notice it's kind of a camel hump shape with a maximum point at some point in the temperature. On either end of the maximum, we're at our low points, essentially usually next to nothing. It makes sense that as we start off at very cold temperatures, the reaction rate is slow. It speeds up as we increase the temperature, but why the decrease? Well, if you remember back to proteins, proteins have a specific shape. As the temperature increases and gets to a certain point, and that varies based on the enzyme, that protein shape starts to break down and change. It's called denaturation. Remember, enzymes have a specific shape in order to interact with their substrate. If the shape changes, it can no longer function and interact with the substrate. And this is what's happening at these higher temperatures. These higher temperatures are causing the protein shape to change, not allowing it to lock together with its substrate well, to the point where eventually it may not function at all. One way you could think of it is well, what happens when you get a really high fever? Why could you actually die from having a high fever? Think about how that affects your enzymes in your body. There are two other things I'd like you to think about tonight. Why do we refrigerate our food? What's the purpose of it? Think about that in terms of temperature and its effect on enzymes. I also would like you to think about why we cook our foods. What's the purpose of cooking our foods with respect to enzymes and chemical reactions? We're going to talk about that in class a little bit more. The second factor affecting chemical reactions that we're going to discuss today is the effect of pH on the rate of a reaction. Enzymes have a specific pH range that they work best at, and that range varies based on the enzyme, based on the organism and the environment that it lives in. Let's take this generic example here. On the y-axis, again, we have our reaction rate increasing, and on the x-axis, we have a pH range of 0 ranging all the way up to 14. If you remember correctly, the closer you get to 0, the more acidic it is. 7 is neutral, 14 is our stronger bases. I have two ranges here. I have a purple curve, which we're going to call enzyme number 1, and I have a blue curve, which we're going to call enzyme number 2. Let's look at enzyme number 1 first. Just based on the graph that you see, what approximate pH range do you think it works best at? I'd like you to try and write that down. The second question I'd like you to think about is, what part of the body might it work in? I'd like you to look at enzyme 2 now. For enzyme 2, what pH range does it work best in? The second question for enzyme 2 is, What part of the body do you think you might find that enzyme in? Why don't you pause, finish answering the questions, and we'll come back and answer them. Now that you've answered the questions, let's go back and see what the answers might be. For enzyme number 1, you see that the peak is the maximum rate. So the highest point of my curve is its best pH range, or pH. So if we look at it, I would estimate that it's somewhere around a pH of 1 to 2 in this case. Well, is that an acid or a base? Remember, if it's below 7, it's an acid. So the pH range that it worked best in was 1 to 2. What part of the body might that work in if it's an acid of 1 to 2? Do you remember what parts of the body are more acidic? If you said the stomach, great job. Our stomach has an acidic pH. We add hydrochloric acid from our glands in order to help this enzyme function well. As the food enters the stomach, it becomes acidic from that hydrochloric acid getting added and allows this enzyme in the stomach to work best. Once that food leaves the stomach, our body dumps additional chemicals into the intestines to make the pH of that content a little bit more neutral or almost basic. If you look at our next enzyme number 2, has a pH range that works somewhere around 7 or so, between 6 and 8, I would say. What part of the body might that work in? Well, most of our body has a pH of somewhere around 7 to 8. This particular enzyme could work in almost any part of the body that isn't part of the stomach. The stomach is really our most acidic part of the body. Think about why that might be true. Why would we have an acidic stomach? What does that help do? That might be something to ponder. We'll talk about that in class. The last factor that we need to discuss in terms of effect on rate of enzyme-controlled reactions is concentration. Now, there are 2 different concentrations we can discuss: the concentration of the substrate or the concentration of the enzyme. Either one will have an effect. The rule goes that as concentration of the substrate or concentration of the enzyme increases, the rate will also increase, and eventually it will level off. As the concentration decreases of either the enzyme or the substrate, the rate will decrease. So if we look at our generic graph again, as the reaction rate increases on the y-axis and our concentration of either the substrate or the enzyme, or it could be both, increases on the x, you notice that as the concentration increases, the rate also increases, but then we get this plateau, it levels off. Why do you think it plateaus? Well, we have 2 questions. What does the rate do as the concentration increases? We just answered that. Make sure you record your answer. The other question is why do you think the rate levels off? Why don't you pause, think about it for a second, and we'll come back and talk about it. Well, we already said that the rate increases as the concentration increases. It can be the concentration of the substrate or the concentration of the enzyme. The reason being is there's more enzyme or more substrate to react, so it can react faster. But why does that rate eventually level off? Well, we have some things called limiting factors. It could be that the reaction just can't possibly happen any faster, no matter how much more enzyme or how much more substrate I add. Think of a factory making, let's say, iPods. If we have a factory making iPods and I add more workers, what could maybe limit them? It might be the speed of the machines. The machines just can't go any faster. Another possibility is that my limiting factor could be I don't have enough materials. I don't have enough machines. In the case of my enzymes, if I increase the enzymes, it may be that there isn't enough substrate for them to react with. On the flip side, if I increase the substrate, there might not be enough enzyme to go around. So the reaction just can't possibly go faster without it. And that's the reason for it leveling off. It's going to maintain that rate, but it just can't go any faster.